fluids, from turbulence to low Reynolds number flows. So I'm going to talk about some of our research about how bacteria behaves in gradients, and to do those studies we use microfluidics. So maybe here it's so we know that bacteria, or motile bacteria, that will be mainly my talk will be only about motile bacteria. They live in complex environments where you have a lot of gradients of different kind. You can have gradients of change in light, change in the magnetic fields of the Earth. You have chemical signals that change in space and in time, and you can have also a gradient of velocities because of shear in the flow. So I will consider in my talk chemical gradients. Okay, it's known as chemotaxy, the way bacteria responds to such gradients, such signals. And I will mainly also talk about how bacteria behaves in gradient of velocity, and this is known as rheotaxy. So I will talk about chemotaxy, rheotaxy, and maybe if I have a bit of time of the collective effects in bacteria. So why this is important? So it's important in many different fields. Here it's one of my interests. It's this situation with my colleague Peter Marguerite from E2BC in Paris Saclay. So the problem is this one: you have this insect, and this insect will eat food, and in this food there is bacteria. So all those bacteria have a population of bacteria. They will move through the gut, so they will have to pass through constriction. They will send some chemical signals, and at the end, in the crypts of the gut, you have only one species of bacteria that will duplicate, grow, and this insect needs this population of bacteria to be able to digest the food. So it's very important for this bacteria, this insect. Maybe the second situation is in the rhizosphere. So the rhizosphere, it's the area close to the roots of the plants where you have a lot of bacteria. And for example, this bacteria, it's soja, so it's what's the name in English? Soja?
Oops.
Soybeans?
Soybeans, yeah. So they emit some chemical signals in the ground, so they can collect one special strain of bacteria. These bacteria will become symbiotic, so it will create a new organ for these plants. It's these nodules here, which are full of bacteria, and those bacteria, they will fix the nitrogen. So they will have carbon sources from the plant, and they will the plant will use those bacteria to fix the nitrogen from the air. Okay, so it's a very important subject because it's if you're able to do that, you don't need to put more introns in the ground. So it's a very hot topic, and so to do that, the plants has to send some signals in the ground. So okay, so this one is okay. So as I said, about 50% of the bacteria swims, and to swim they are using flagellas. Okay, so it's an assembly of proteins. Here it's an example of flagella, so it's helicoidal. It has an helicoidal shape. It can be right or left-handed. Here it's examples of different type of bacteria. So you have the body of the bacteria, so you have a membrane on the bacteria, and you have and the flagellas are connected to the membranes through nanomotors, and the motors can rotate in different directions. So you have different. So for example, I will mainly talk about École. So École has like 5 to 6 flagellas. They are a little bit everywhere around the membrane, and but you have different cases. Here, Pseudomonas, they have only one single flagellas at the polar here, and you can have different way of putting the flagella. So how it works? Here it's an example taken from Berg's labs. So the typical length. So here, this size, it's like 4 to 5, let's say 4 microns, 1 microns here, and if you include the flagellas, it's about 10 microns. Here on the movie, it's you will see they were able to taint the flagellas and the bodies, and you will see how the bacteria use the flagella. So here you see when all the motors turns in the same direction, all the flagellas they make a bundle, and so they rotate in some direction, and it's how you will have propulsions of the bacteria. After a typical time of 1 second, that's the case for École, 1 or 2 or 3 motors, they start to turn in the other direction. So they change the direction of rotation, and it's the reason why you see that you have unbundles. You see here, so you see the flagellas, they unbundled, and this induces a rotation, a change of direction of the body of the bacteria. Okay, so we said that for École perform runs, okay, with the typical velocity of 20 microns per second, and tumble every 1 second. So it's like a random walker. Okay, it performs random walks in a fluid. So I will move to the first part of my talk, is how we can study, how we can use microfluidic to study bacteria in a porous medium. Okay, so it's very relevant. So in many different kinds of different fields. In soil, we just saw with the last talk, it's also important to move, to use this to understand how cells moves in capillaries. And I will talk about rheotaxis, so it's how we can understand the coupling between bacterial motility and the flow heterogeneity we have in typical porous medium. And I will mostly talk about dispersion. So dispersion in the porous media, it's the following phenomena. Imagine that this is the scale, so this is grains, okay, that are randomly placed, okay? And if you have a flow through this network, you have a very heterogeneous flow structures. So the flow velocity will be very small on the grains, and maximum between the grains. So if you put tracers or objects at the inlet, and you look at where are those objects after a certain time, you will observe that you will have dispersions of the population. So some objects will have traveled far away inside the porous medium, and some will have a very small displacement. And if you look at the distributions of those objects after some time, you will see that there is a maximum probability here, which is just the average flow rate or average flow velocity times the time t, and you have dispersions of the population. And usually, this dispersion here grows like the square root of time. Okay, and that's the typical picture we have for porous medium. And our aim is to say, okay, let's put now motile bacteria. What will we observe? Will we observe this type of probability distribution? Will we recover this type of square root time typical law? So, the problem is when you want to use porous medium, it's difficult to see inside or impossible to see inside. So what we have done is we were able to reproduce using lithography, classical lithography. We reproduce the heterogeneous structure of the pores of the porous medium by putting circular obstacles, okay, cylindrical obstacles inside a chip, okay, a microfluidic chip. More precisely, here we have. So here it's the cell we have. So the width of the channel is 500 microns. The height is 100 microns. And the typical size of the obstacles is 40 microns. And they are randomly placed into these channels. And the good things with PDMS, it's so we are able to reproduce such structures. It's transparent, so there is no problem to be used under a microscope, and it's porous to gas, to oxygen. So it's very nice because if you have bacteria inside the chip, it will have a sufficient amount of oxygen going through the PDMS so that we don't have any motility problems in the experiment. So it's here's the microscope.
Sorry to interfere and stop you at the beginning. Those structures are the static structure made, or those are part of the structure?
Yeah, they are static.
Static, okay.
They are static.
Okay.
Yeah.
Micropillars, short of micropillars?
It's micropillars of 100 microns height, so they are quite high. And so we use 100 microns to avoid too much 2D effect in the flow. Yeah. And also because, like you will see, bacteria, they love surfaces. So we put 100 microns so to have bacteria that are far away from the two surfaces. And here it's the typical flow velocity we have in the chip, in the device. So here you see it's obstacles, okay? And we were able to reproduce one property of porous medium, it's channelization of the flow. So you see you have channels where the flow velocity is high, okay? And so you can see here it's a typical channel, so you have a strong channelization of the flow. And you have regions where the flow velocity is very small, okay, it's almost zero. So you have this this is a typical flow that we have.
Is this simulation or?
No, that's experiments. Okay. You put tracers in the flow and you track the tracers and you do that. Here it's so if we have what we are able to do, okay, is to record to do some movies, and from the image treatment of the movies, we are able to track bacteria or tracers and to obtain the trajectories of the object. So we are able to, like that, to have the flow velocity in the flow cell. And now we will track bacteria, and we will look at the properties of the bacteria. So we use two types of bacteria, motile École bacteria and non-motile bacteria. Okay, and you see all the lines is trajectories that we record. Okay, that we from the movies we were able to extract all those trajectories. So here it's when the bacteria are non-motile, so you see they go through the pores, almost like in a straight, they go through the pores in a straight way. When the bacteria are motile, you see it's a bit more complex. You see bacteria able to leave the flow, the channels, and to go and visit the pores. So we done the following analysis. So like I said previously, you have all the positions of the bacteria. So you look at all the population spread in the pores. So you have the flow, they are transported by the flow, and you look at where are the bacteria after 1 second, 3 seconds, and 5 seconds. And so you see, that's blue, it's after 1 second, red after 3 seconds, and yellow after 5 seconds. And we were able to fit those distribution by a Gaussian distribution. So for non-motile bacteria, everything behaves like what we expect, that's the classical view of what happens in porous media. So you have dispersion, so you have a spreading of the population of bacteria by the heterogeneous structure of the flow. Now, if you do that with motile bacteria, what you observe is now, after 1, 3, or 5 seconds, the maximum probability of the distribution is at 0. So meaning that and you have a spreading like that. So you have with time you have more and more bacteria inside the porous medium, but a lot of bacteria they just stay at the positions where you observe them. And this is because if you look in details about some of the trajectories, you will observe that the bacteria are retained inside the pores. Okay? And they need motility to be retained inside the pores. So to understand why they are retained inside the pores, we can use those. I will show you one movie. It's maybe a bit. So here you see grains. That's grains. So it's a zoom inside the flow cell. So you have the grains, and each white cylinder is a bacteria. And you have the flow, and you see here the flow is very strong. So they move like that. And you can see that they are able to leave the flow, and then if they leave the flow, they go behind the grains. And in the grains, some are swimming along the surface of the grain. So you see here, and we go back to the flow. So they leave, you see, they leave this one, for example. So here you see that they leave, they move behind the grains, they move along the surface, and they go back to the flow. Okay, and it's because of this effect that you have this retention inside the pores. And here the flow it's only 35 micrometers per second, so it's a very low flow rate. There is no it's low Reynolds number flow, and it's about 2 times the swimming velocity. And we were able to do experiments that show the same. And here you see you have a grain, a single grain. You have a flow of bacteria, and you see you have an accumulation of bacteria behind the grains. And we are able also to observe this using numerical simulation. So we developed some models to understand, or to not to understand, but to upscale our observations at macro scale, so that can be so that we have tools to be used for large scale porous medium. So just a comment on this curve of these two curves. So here it's the normalized velocity distributions of the particles when they are non-motile. So you see each symbol it's a different flow rate, and you see if you normalize the velocities of the particles, all the distribution collapse. And now when the particles are motile, you see that you have an extra number of bacteria that have low velocity and less bacteria that have high velocity. So it's a way to quantify just the observation. So we develop we upscale our observations, so we were able to have effective coefficients that can be used for large scale simulation or observation. And okay, it's one of the outcome of the model. It's what we call the retardation effect that decrease when you increase the flow rate. And this model is just is based on the idea that we have the population of bacteria can be separated into bacteria that are in the fluid, okay, transport with the average fluid velocity, and a fraction of bacteria trapped into the low flow regions, and we can have a typical time of exchange between the two families, and this translates in terms of retardation, which is here. So you have a strong retardation when the flow rate is low, and almost no more retardation at large flow rate. So that was some of our work on the effect of rheotaxy on the transport of bacteria. I will move now to the effect of chemical gradients on the motility of bacteria. So, like you know, I guess it's so here it's a sketch of the bacteria. Okay, so you have the membrane, you have the motors, you have the flagella, and on the membrane you have also some receptors, and those receptors, they can bind with some chemicals. So for École, you have different, you have 5 chemo receptors that have been identified, and they all bind with different type of chemicals. So one of the challenges is to, for École, the picture is very clear. It's known since everything is quite known since many years. but the issues, it's for less study species of bacteria, where it's difficult to understand which chemoreceptors will react with different chemicals. So the idea is to have a platform that can be used to easily identify chemoreceptors on chemicals and know if we have the right combination of receptors, chemicals, giving rise to chemotaxis. What's the physics behind chemotaxis? It's very simple. In fact, you have here the chemoreceptors where some chemicals bind. You will have a loop, a very complex loop that I'm not going through details, but all this loop will influence the probability for the motors to switch direction of rotation. Okay, so if the environment is good, if you have a lot of, for example, good molecules that bind to the chemoreceptors, the probability to change direction will be low. Okay, so the bacteria will continue to move in the direction, in the straight direction. But if this change, then the probability to change direction, so because, for example, if you go down the gradient, so you have C that decrease, that will influence these motors, and the bacteria will switch direction, and so we'll go back to the good direction, which is a gradient that increases. Okay? So, in fact, bacteria, they sense gradient because they move inside the chemicals. And so, as I said, they have a swimming velocity, and the effect of chemicals will introduce an additional velocity, a chemotactic velocity, that is named VC. This chemotactic velocity is proportional to the gradient of concentration. And you have here, P of C, it's the susceptibility of the bacteria to these chemicals. And it's a non-linear function of the concentration. So the point is to be able to measure that. That was our goal, to measure with precision this function. And the difficulty is that usually this drift, chemical drift, is about 10%, maybe less, sometimes less, maybe 15% of the swimming velocity. So it's challenging. It's challenging to be able to measure this drift. So here it's the setup we used. Okay. So we have here we use a 3 channels system. So you will see it a bit better after. But the idea is we have 3 microfluidic channels. The size of one channel is 600 microns. The height is 100 microns. And so you have, that's the channel in the middle. You have channel 1, channel 3. And the idea is to put chemicals in these 2 channels with different concentration. So we have different concentration here and here. And the system is placed, these PDMS, those PDMS channels are placed on the top of agar layer. And so the chemical, they can diffuse through the agars, and by diffusion you will create a gradient between the 2 chambers. Then you will put bacteria in the middle. Okay, and we will observe with the microscope, and if we are correct, if they are attracted to these chemicals, we will see that the population with time will move towards the source. Okay? And the nice things with this system is that we have a stationary and controlled gradient. Okay? The gradient is constant. Okay, here it's a typical experiment. We put alpha methyl aspartate in this channel. We put the buffer here, and we in the middle in this channel we have a suspension of bacteria, École, and there the bacteria is fluorescent. So each dot it's a bacteria. And you see, this is time. Yeah. Okay, so it's. I will play it again. So in the beginning, you see they are everywhere, and here it's minutes, and you see after 40 or 25 minutes, 1 hour, you see all the bacteria are located in that regions. Okay, and so it's really you see there is a chemical attraction of the bacteria towards the chemicals. Here it's 3 experiments. The first one it's what we observe when there is no gradient, okay, and I should mention, since we are recording movies, we are able to track, to analyze each bacteria trajectories, and all those lines are tracks of bacteria, okay. So here there is no gradient, okay, no chemicals, and you see that there is no effect. And you can look after 5 minutes, 25 minutes, no effect. Now, if you put a gradient, you see that after 25 minutes, it's exactly what you saw on the movies. You have bacteria moving toward the source. Here it's movies recorded on the surface. On the agar surface. So on the agar surface, you see 2 things. First, you see the trajectories of the bacteria. They do circular trajectories. Okay, but so they rotate like that. But nevertheless, you see after sometimes they move where you have the attractants.
So what is the color legend? Because you are showing in time. So
Yeah.
0 minute, 5 minutes. So this is again time.
Sorry, large T, it's time 0. Then we record a 20-second movie or 25 second movie at T0.
This right side, that 25 is.
That's the time in one movie.
Oh.
And after 5 minutes, we record a new movie. Okay, and that's a 25-second long movie. So the color code of this is this, and that's large T. It's the time since the beginning of the experiment. So I'm skipping all the details. We developed a way to analyze the trajectories to obtain the chemotactic velocity, chemotactic drift. So the chemotactic drift, it's the balance between the velocity of the bacteria that we have to correct by the fact that you see that you have an accumulation of the bacteria. So the population, it's you have a profile of a concentration in bacteria, and this induces diffusion. Okay, so we correct this flux, convective flux, by the flux induced by diffusion. Okay, so that's what we developed, and so we can obtain the chemotactic drift, and from the chemotactic drift, okay, we are able to obtain the susceptibility. Okay. Here it's just a set of experiments where we change only this value. We maintain the average concentration of chemicals constant, and we change the gradient. Okay, so we basically what we done is the C is constant, and we change the gradient. So we are doing something like that, and you see that the drift increase with linearly with the gradient of concentration. So that's the first check. Ouais.
On a la salle à partir de midi, normalement, c'est pas ici. Vous êtes sûr? Non, c'est réservé pour la journée. J'ai le film pour la conférence. Ok, bah, merci de l'émission. Normalement, c'est réservé pour la journée.
Okay, so when you see here it's normalized. We normalize the drift by the swimming velocity, and you see the maximum. It's the chemical drift, the chemotactic drift. It's 25% of the swimming velocity. It's why it's tricky, because the drift it's a small part of the swimming velocity. And here it's the chemotactic susceptibility. So you have here the concentration in alpha-methyl aspartate. And in blue, it's all the small dots, it's our results. Here we average our local observations, and we obtain these blue circles, and we compare our method with a previously developed method that analyzed the profile in bacteria. And so you see, so we were able also to obtain the range of chemical concentration where you observe this chemotactic drift, and you have here you see that you can in that range fit the susceptibility by 1/C. So this means that the chemotactic velocity goes like gradient of C divided by C. So it's log of C. So it's the typical log sensing. That it's classically observed, reported in the literature. We also applied this to aerotaxy. Aerotaxy, it's instead of a gradient of chemicals, it's a gradient of oxygen. It was very difficult to have a setup where we can control really the gradient of oxygen. So in that case, in that experiments, the gradient of oxygen is induced by the bacteria themselves. So in fact, we put the bacteria in a channels where here it's sealed, here it's open to oxygen because we have PDMS. So the bacteria, they will consume oxygen, so they will create a gradient themselves because they consume oxygen, and they will move in that gradient. And we were able to measure the local gradient of oxygen because we have we use a special ruthenium dye that is able that is sensitive to oxygen. So we have all the information, the local gradient of oxygen, we can track the bacteria, have all the local velocities, so we can apply all I said just before. And here it's the susceptibility of the bacteria. Here it's the C, it's the oxygen concentration. And here in that case, you see that the susceptibility goes like 1/C to the power 2, which is different than from what we observe with chemicals. And so now so I don't know how long. 5 minutes? Okay. So we also done some experiments where we now, instead of having few bacteria, we have a large number of bacteria. We want to understand what will happen. So when you have a family of bacteria, a dense population of bacteria, you will develop collective effects. So that's known since on study since many years. The collective effect comes from the fact that when the bacteria moves, swim in the liquid, it also displaces the liquids. Okay, so it pushes. Okay, so you have a flow field around the bacteria. Okay, it pushes the liquid to move. Okay, so and to move, it has to push the liquid ahead, and the flow lines are like that. Okay, so you have liquids going like that, liquids going like that. So you can see that like it's a force dipole. Okay, so you have some hydrodynamics. So when two bacteria are close, because of the flow field generated by the swimming activity, you will, because of the flow field, they will start to align, okay? And this will be a source of collective motions. What is nice also, it's when they swim close to a surface, this will be will act like an attractive forces. So it's why you can explain why bacteria, they like to stay on the surface. And moreover, because the flagella rotates, the body also rotates, okay, in the other direction, because it's low, because of torque conservation. If this rotates in this direction, the body rotates in the other direction, and because the bacteria moves, it will create circular trajectories like I showed you previously. Okay, and we know that this will generate what we call an active stress, an additional stress in the fluids, which is active stress. And in the case of pusher, like École bacteria, this active stress will affect the effective viscosity of the liquid by lowering, it will lower its viscosity. And it's what we were able to measure here. It's the viscosity of a suspension of bacteria as function of the shear rate. Okay. Here you have non-motile bacteria. You see non-motile bacteria, there is no effect on the viscosity. And when you have motile bacteria, you see you have a drop of viscosity. So it's due to this additional stress coming from the swimming activity. Okay. It's only true when the shear rate is small, okay, because if the shear rate is too high, then you flush the bacteria and there is no effect on the viscosity. So typically you have to use a flow that is about swimming velocity, okay? Okay, so So you put bacteria, you are able to decrease the viscosity. You can go to zero, okay? Zero viscosity. It's when you have collective effect. So with one of my students, Akash Ganesh, he had this wonderful effect. Idea is to say, okay, I learned that if a less viscous fluid push a more viscous fluid, there is like a submental instability. So I will, he said, okay, so that's known since many years. So let's put bacteria and push the same liquid without bacteria. If I'm right, I will have a submental instability because bacteria will lower the viscosity. So he developed a setup based on using DMS. I'm not going through all the details. So if you have a small number of bacteria, so here you have the fluid with the suspension with bacteria. Here you have no bacteria, and you see the front is stable, okay? Because you have a small number of bacteria. In that case, it's quite dilute, so the flow is stable. But now, I don't know why it's not starting, but maybe this one. Too slow. I don't know. You have to believe me. So, but you see your fingers in the beginning, and you have at the end you have a single fingers that moves into the liquid, and it's very funny because the particles are here. So Einstein said if you put particles in a liquid, you will increase the viscosity, but it's not really true. If you put bacteria, you put active stress into the liquid that will lower the viscosity and that will induce this instability. And we were able to look at so the front, the width of the finger as function of the number of bacteria. And we say you have no fingers, and when you cross this moments where you start to have collective effect, you have the finger. Okay, with that, I will just conclude. Microfluidic is a really wonderful tool to study motile bacterial motility because you can really control the environment, control the flow, you can track bacteria and the population of bacteria, and it's a wonderful tool to play. And it's in couples of years we were able to make all these new discoveries about rheotaxy, chemotaxy, etc., etc. And it just opens new avenue to new research. And it's funny that you can play with the property of a liquid by adding active elements. So all this work has been done in collaboration with people from my lab, Frédéric Moisy, Karim Douarch, Prof. Jarawi, some PhD students, Julien Bouvard, Akash, Adam Akrepi, and some collaborators from different labs, Eric Clément, Marc Odens, and Peter from EPC, and Veronica Angelo from Buenos Aires.
Long live India and France friendship.